So today we are going to do the third part of the chapter pollination and fertilization. Let's start with our slides. Pollination by humans. This is a topic that I have missed in the earlier part, so I would like to continue with it. It is known as mechanical pollination or artificial pollination. Definition and requirement. It is a manual transfer of pollen from the male stamen to the female pistil. Okay, the same thing, the same transfer of pollen from the stamen to the pistil, but only it is a manual transfer. And improved crop yields in spite of a reduction in the populations of pollinators due to the use of these fungicides and other chemicals, pesticides and other chemical fertilizers. You can say there are great strike at the life of these pollinators, so their numbers are decreasing, and the numbers of the poor yields of crops are increasing. To avoid this dominance of the poor crop yields, this artificial pollination is done so that there are more and more improved crop yields. That is, the pollination takes place in spite of the less number of pollinators. What are the methods? The first method you can see, that is the simplest method uh, of the hand pollination technique, is simply to shake the plant. Okay, it is mainly productive or effective in plants which produce hermaphrodite flowers. Hermaphrodite flowers. Hermaphrodite flowers means the bisexual flowers. So if you are just sucking the plant, so the pollens will get transferred from the anther to the pistil. Okay, they might get, and most of the cases they do. And to hand pollinate squash, pumpkins, lemons, and cucumbers, that is the cucurbit family, you have to pluck the petals of the male flower and use a small paintbrush or cotton swab to transfer the pollen to the pistil. You can see the picture here. See the picture here. You can get that they are using a brush to take the pollen, and then they are just transferring it to the pistil. Okay, you have seen this one. Nice. Now we are going to do the fertilization in plants. First thing, you need to know what is fertilization. It is defined as the fusion of male and female gametes to give rise to a new individual, no matter it's plants or animals. So it is mainly the fusion of male and female gametes to give rise to a new individual, no matter it is any plant or any animals. Because this is a basic definition, but here we are going to do the fertilization in plants. Okay, that's mainly in the flowers. So types of fertilization, so it can be grouped into three and is classified based on the entry of the pollen tube into the ovule. First one is porogamy. It is a common type of fertilization which is carried out in all angiosperms, that is the flowering plants. And in this type of fertilization, the pollen tube enters the ovule through the micropyle. Through the micropyle. Okay, micropyle is the opening of the ovule. And then the mesogamy. It is mainly seen in the cucurbit family, such as pumpkin, ridge gourds, bitter gourds, and other gourd plants. In this fertilization, the pollen tube enters the ovule through the middle part or the integuments of the ovule. Integuments of the ovule. The next one is the classogamy. This type of fertilization is seen in Cosorina species. Cosorina species, and in this condition, the pollen tube enters the ovule through the classa tissue. Classa tissue. You can see the picture here. The first one is porogamy, the second one is classogamy, and third one is mesogamy. So micropyle, porogamy. Micropyle has P and porogamy has P. So you can remember in this way. Classogamy with the classal tissue, you can just remember it. And mesogamy. How you remember it? Mesogamy is through the middle portion. See, you can see the middle portion. So middle M is there, for mesogamy M is there. Okay. Next we will see the double fertilization. Double fertilization is a process of fertilization characterized by the fusion of a female gametophyte with two male gametes. that is the fusion of a male gametophyte with two male gametes. In this mechanism, one sperm fuses with the egg producing zygote and the other fuses with the two polar nuclei to make the endosperm. So if I could highlight it, so I could highlight this point, that is one sperm cell fuses with the egg producing zygote and the other fuses with the two polar nuclei to make the endosperm, and all angiosperm undergo double fertilization. Done? Okay. We'll see the structure of a pollen grain. After pollination, pollen grains are deposited on the stigma of the carpel. Under suitable conditions, pollen grains produce a tube, which is known as the pollen tube, and this tube goes down to the stigma and style towards the ovary. So you can see the picture here. See this portion. This portion is the vacuole. The outer layer is known as the exine, and the inner layer is known as the intine. This is a jumbo. Okay, this is a generative cell. This is a tube nucleus, and this is a tube cell. Have a look at it. Okay, now you'll see the germination of pollen grains. In flowers, pollen germinates after the pollination of the carpel and grows into the style by creating a pathway from the pollen grain to move down to the ovary. In flowers, the pollen grains germinate after the pollination of the carpel. Carpel means here the female part, and grows into the style by creating the pathway from the pollen grain to move down to the ovary. And this pathway is the pollen tube. Remember it. It's the pollen tube. The pollen tube breaks into the ovule through the micropyle and bursts into the embryo sac. So the pollen tube breaks into the ovule through the micropyle and bursts into the embryo sac. Here the male nucleus fuses with the nucleus of an egg inside the ovule, forming a diploid zygote, which later swells and develops into a fruit. So the male nucleus now fuses with the egg inside the ovule and forms the diploid zygote, which later swells up and develops into a fruit. Okay. Now here you can see one picture. You can see here. See, here is the pollen grain. So it is going through this pollen tube and it is just entering this ovule. This is the ovule. Okay, and this is the ovary. Please remember it. The double fertilization takes place in the ovule, not directly in the ovary. Okay, inside the ovary, in the ovule. Okay, done. Okay, I know that you are thinking that why I have changed the slide. You'll see I have more nice pictures to represent this one. Now, what happens in double fertilization? The nucleus of the pollen grains divide by mitosis to form two male gametes. So the nucleus of the pollen grains divides by mitosis. Remember it, by mitosis, and forms two male gametes. The tip of the pollen tube after entering ovary discharges the two male gametes into the embryo sac. So after entering the ovary, the pollen tube discharges the two male gametes into the embryo sac. One of the male gametes fuses with the egg to form the zygote. Okay, wait, I have done a mistake. Okay, so one of the male gametes fuses with the oh, so one of the male gamete fuses with the one of the male gamete fuses with the egg to form the zygote. This fusion is known as fertilization. Another male gamete fuses with the diploid secondary nucleus and forms the endosperm, a food storing tissue. Okay, so what happens? One of the male gametes fuses with the egg to form the zygote, which will later develop into the uh, other stages that will come later. This is known as fertilization. And the other two male gametes fuses with the diploid secondary nucleus. Sorry, other one. There are two male gametes. One of the male gametes is fusing with the egg, and other male gamete is fusing with the diploid secondary nucleus and forms the endosperm, a food storing tissue. Okay, here you can see the picture. See, here you can see the exine, the intine, the tube nucleus, the generative nucleus, the jumbo. See, here it is just starting. The pollen tube is just starting to generate. Here these two are the sperm nuclei and this is the tube nuclei. Okay, you can see the pictures to just get it. So these are the antipodal cells. Please remember it. Antipodal cells are opposite to the micropyle. Okay, these are the synergies. This is egg cell, micropyle, embryo sac, polar nuclei. This will be nuclei. Okay, nuclei, not nuclei. Some mistakes are there. Don't, please forgive me for that. Okay, okay. So you can see the central cell, the integuments, the pollen tube, the micropyle, the egg cell, the embryo sac, the polar nuclei, the nucleolus, and the ovule. This is the ovule. Okay, and if I draw a bulging structure outside, this is the ovary. Okay, don't tell that the double fertilization takes place entirely in the ovary. It takes place in the ovary, inside the ovary, in the ovule. Okay, okay. Now we'll go to the next slide. See here. See here very clearly. You can see the image here. See, this is A. So A, you can see the germination of pollen grain. Okay, so it is germinating. B, you can see that the tube has already formed and it is entering the ovule. Okay, it is entering the ovule. B have seen. Now see, from C we are now seeing the closer look of the ovule. So the pollen tube is here. Now see, these are the two male sperm cells or the male gametes. Okay, this is the haploid egg nucleus. Here you have the binucleus cell with two haploid nuclei. Okay, two haploid nuclei is this one. These two polar nucleus are there, and here is the haploid egg nuclei. Now see, C is done. Now D. Please have a look here. D. The haploid nuclei. See, from here they have now transferred the two eggs. Transfer the two male gametes into the ovule. So this is the haploid sperm nuclei, and here one haploid sperm nuclei has already fertilized. It is getting fertilized here with the egg. It is getting fertilized here with the egg, and here with the two polar nucleus again it will get fertilized. If you are not able to understand, I will again show you. Please wait. Please wait. Let me use this color. So here they are again getting. This is the male nuclei. This is the female to polar nucleus. So it is getting fertilized here, and here with the egg nuclei one is fertilized. And finally, here the diploid endosperm cell is formed, and here the diploid zygote is formed. Here the diploid zygote is formed. Got no? You can see the picture once more. I will remove all the ink. Okay, just see here. A, B, C, D, E. Okay, I'll just remove all the ink so that you can understand this picture once. I have just removed all the inks. You can now see it. Please have a look. At the A, there is the germination of pollen. B, the pollen tube has already grown and it is going to the ovule. Okay, next you have seen the closer look of the ovule now. Now the two sperm cells are arrived at the opening of the ovule. Done. Okay, you can see that here we are having the binuclear cell. Here we are having the binuclear two haploid nucleus there, and here we are having one haploid egg nucleus. Now one sperm cell is going, that is one male gamete is going and fertilizing with this egg cell, and the another one is going with these two, fertilizing with these two polar nucleus, and finally The polar nuclei, that is the two polar nucleus and one male gamete is forming the diploid endosperm cell. This is known as triple fusion. The phenomenon of the fusion of one male gamete and the two polar nuclei is known as triple fusion. Triple fusion. Triple fusion. And here one male gamete has already fertilized with the egg cell. This is known as syngamy. Okay, this is known as syngamy. I will write it for you all. Please wait. I will write it for you all. This is a triple fusion we have seen, and this is known as syngamy. Wait, I will just remove all the colors and again I will write here. This portion is here the fertilization of the egg and the male nuclei or the male sperm cell is known as the syngamy. Syngamy. Okay, fine. Next, what happens to the different parts of the flower after fertilization? Are they still present or they get vanished? Let's find our answers. See the chart here. You can see that we are having sepals which usually wither away, but in some cases they remain attached to the fruit. They're like the brinjal, tomato, etc. Next we are having the petals. Petals wither away, stamens wither away, styles, stigma wither away. Okay. So if I could give it a one thing, so like this, it would be right. Like this, it would be that these all wither away. Petals, stamen, style, stigma all wither away. Sepals may wither away or may not be, as it is present in some fruits like the brinjal, tomato. Although they are the vegetables, tomato is often considered as fruit also. But here we are just discussing the development, so we'll consider it as the fruit only. Okay. Next, basically, we don't go to classify them as vegetables. We just take them as the fruit. Next, we are having no need to go that with the controversy of is it a fruit or vegetable, but just understand that how it is developed. Okay. Then ovary, it becomes a fruit. Fruit wall, ovary wall will become the pericarp. Ovule will become the seed. Placenta will become the stalk of the seed. Okay. Then the outer integument will become the testa, inner integument will become the tegmen. These two will form the seed coat. Then the secondary nucleus will become the endosperm. The egg cell and the synergy will become the embryo, and the antibodal cells will disorganized. Okay, will get disorganized. Okay, so you are done with it. So you can see the face of the floral parts also. Okay, now I am going to show you a picture. Some videos I am going to show you. Please wait for it. Please wait. Please have a look at the video.
Flowering plants undergo a unique reproductive process where there are two fertilization events. This double fertilization event occurs between the male reproductive organ, the male gametophyte, and the female reproductive organ, the female gametophyte. Before the fertilization event can occur, the ovule has to undergo some changes. At present, the ovule contains one reproductive cell, known as the megaspore or mother cell. This cell is diploid and undergoes meiosis, producing 4 haploid megaspores. In the majority of species, 3 of these megaspores degenerate, leaving only one surviving megaspore. This surviving megaspore expands and undergoes 3 rounds of mitosis to produce 8 haploid nuclei. As the nuclei have not developed any individual division, they initially share the same cytoplasm. opposite the ending of the ovule, known as the micropyle. Another 3 cells form above the micropyle. 2 of these are synergids, and the other is the egg cell. This leaves 2 nuclei in the center of the ovule. These central nuclei remain together in one large cell. It is the egg cell and this central nucleate cell which will eventually become part of the double fertilization event. In order for the double fertilization event to occur, the male gamete, the sperm, must travel from the anther to the embryo sac within the female reproductive organ. The pollen grain contains 2 main cells. A cell named the tube cell makes up the bulk of the pollen grain, and the sperm cell, which at this stage is known as the generative cell. To reach the embryo sac, a pollen grain must land on the stigma. Once landed, it begins to germinate. The tube cell forms a long structure down the style and into the ovary. The generative cell travels behind the tube cell nucleus. Once near the ovary, it divides by mitosis to produce 2 haploid sperm cells. The pollen tube reaches the micropyle and releases the sperm cells into the embryo sac. One of the 2 sperm cells fertilizes the egg cell. This produces a diploid with sulfur, where a diploid zygote.
Okay, so we have seen that how it is how the pollen grain is getting germinated and how it is entering the ovule. We'll see one more video to see a more closer look of it. Okay, please bear with me once. Please bear with me.
S.G. Nauschen, 1898, was the first to show that both the sperms released by a pollen tube are involved in fertilization. They fertilize 2 different components of the embryo sac. The phenomenon is unique to angiosperms and is called double fertilization.